Hello everyone at Medical Sciences. In this session we are going to look at what we call the structure of a bacteria. If it is your first time on this channel, please you are requested to subscribe. Like the video when you enjoy it and share to many others so that we can continue to grow. So on the structure of bacteria, we know that bacteria is a prokaryotic cell which is unicellular. It is prokaryotic cell and it is unicellular organism with no nuclear membrane and lacks cell membrane bound organelles. So we want to see different parts or structures of this unicellular organism we are calling bacteria. And the bacteria is made up of, we are going to divide them into two. We have the outer parts and the inner parts. And among the outer parts, we are going to see what we call the capsule. So we are seeing the most outer portion is the capsule. But some bacteria, they don't have the capsule, but they have what we call the slime layer. So some can have the capsule or slime layer. Slime is more of viscous than the capsule, but all of them serve the same purpose. Then from the capsule, we see presence of the cell wall. So it is followed by the cell wall. So all this in black is known as the cell wall. So this is the cell wall of the bacteria. Then from the cell wall, as we go inside, we find what we call the plasma membrane, or you can call it cell membrane. Its name is either cell membrane or plasma membrane. Then on the cell membrane, we have a hair-like appendage which is very long, attached to it with the basal body, and this one we call it a flagella. So this is named as flagella. When it is one, you can call it flagellum, but when there are many, they are flagella. So this one, since it is one, we can call it a flagellum. Then from the flagella, we have also these short appendages which are attached on the cell wall. And these ones, we are going to call them pillars. When it is one, you can call it pillars. When there are many, you call them pili, or sometimes you can call it the fimbriae. So these are the capsule, the cell wall, the flagellum, and the pillars. In their combination, they form what we call the outer membrane, or the outer part of the bacteria. Whereas the inner part begins with the cell membrane, which we are calling the plasma membrane. And within the cell membrane surrounds what we call the cytoplasm. So the whole area inside this cell is known as the cytoplasm. So we are going to see all this is the cytoplasm. And this cytoplasm is the one that we find all these organelles embedded within the cytoplasm of the cell. And this cytoplasm contains bodies like ribosomes. We're going to see ribosomes. These are ribosomes. This is a ribosome, this is a ribosome, and this is a ribosome with a messenger RNA. Then from the ribosome, we also have what we call chromosomal DNA. This is chromosomal DNA. And this chromosomal DNA is always located within what we call the nucleoid. So the whole of this area where the DNA is located is what we call the nucleoid. So the chromosomal, which is the coiled DNA, is found within a region known as nucleoid. And we have said that this prokaryotic bacteria, it lacks the nuclear membrane, but it has what we call the nucleoid, which is the location where it is found. Then we have also other components like the cytoplasmic inclusions. This is cytoplasmic inclusions also present within the cytoplasm. We have also the endospores. Endospores are found in some bacteria like the Bacillus anthracis. You can find them also in Clostridium species like Clostridium tetani. So these are some of the two examples of bacteria which have spores within their cytoplasm. So these are the parts, and now in our session we are going to see the function of each part of the bacteria. And we are going to begin with the capsule as number one. So number one, we are going to begin with the capsule, which we see that some bacteria have what we call a slime layer, but doing the same function. And this capsule is the outermost part that surrounds the bacteria. And their role is they offer what we call virulent factors by preventing, they act by preventing phagocytosis. That bacteria which possess capsules, because being slippery, when the phagocytes like the macrophages and the neutrophils within the host body are trying to engulf this bacteria, it becomes slippery and hard to be engulfed due to presence of this capsule. And apart from that role, it also prevents the bacteria from drying out. So the bacteria cannot dry out due to presence of the capsule. At the same time, it protects the cell wall. This capsule protects the cell wall of the bacteria from like antibiotics or from destruction by the surrounding areas. And apart from these three functions of the capsule, we can also use capsule for bacterial identification, whereby we can use like the capsule antigen to identify organisms which have capsule from those ones which are non-encapsulated. So these are some of the functions of the capsule of the bacteria, or what we call the slime layer to some bacteria which have the slime layer instead of the capsule. So this, in case we ask to outline any four functions of the capsule, you should be in a position to tell us some of the functions of the capsule. After looking at the capsule, we now want to look at the cell wall, because we have started with the outer parts of the cell wall. So we are on the outer parts of the bacteria, whereby we have begun with the capsule. So number two, we can look at the cell wall. And this cell wall of the bacteria is made up of what we call peptidoglycan layer. It is made up of peptidoglycan layer. This peptidoglycan layer is made up of proteins and the glycans, which we call the carbohydrates. Those are the polysaccharides, whereby it is made up of the N-acetylmuramic acid and N-acetylgalactosamine. And these layers differ from the gram-positive, have multiple layers of peptidoglycan, whereas gram-negative have single layer of the peptidoglycan. And that's how we are able to distinguish them by their gram-staining reaction. And the function of the cell wall on the bacteria, one of it is to provide shape. It provides shape and rigidity to the bacteria. So it is the one that makes the bacteria either to be a cocci, to be a bacilli, to be comma-shaped, to be spiral-shaped. So that shape is provided by the cell wall of the bacteria. Another thing, this bacteria is able to protect the cytoplasm or the cell membrane from osmotic stress. Because when there is creation of higher osmotic pressure, so what prevents this bacteria from bursting is the presence of this rigid cell wall made up of peptidoglycan layer. So we are going to say that it prevents cell from bursting due to high osmotic pressure. So when there is a buildup of osmotic pressure within the cytoplasm, it is prevented from bursting by the presence of the rigid cell wall. And apart from these two functions, we are going to see that this cell wall also is permeable. And being permeable, it enables entry of materials within the bacteria. That is another thing. So it is being permeable. It enables entry of materials within the bacteria. Then another thing is it provides or it anchors the flagella and the pili. So you can see that this flagella is anchored by the cell wall. I told you the cell wall is in the black color in the middle, whereas we see even the pillars and all the fimbriae, they are also attached to the cell wall. Hence, it provides the support to them. So these are some of the functions of the cell wall. And these cell walls also enable us to identify bacteria. For example, due to gram-stain reaction, that if we find this one is purple, we know this is gram-positive. If we find another bacteria is red or pink, we can know that this is gram-negative. So it can also enable bacterial identification and typing, identification stroke typing. So these are some of the functions of the cell wall we are talking about under the outer parts. So from the cell wall, let us briefly look at the flagella. So part number 3 is the flagella. And we said if it is one, we call it flagellum. And some bacteria, we are seeing the flagella being a hair-like structure, but longer than the pillars. And these flagella, their function is to enable movement. They enable movement, or what we call motility. They enable bacteria to move from one place to the another. So bacteria which are non-motile, they lack the flagella. Whereas those with the flagella, they are able to move. So this one enables us to classify bacteria from those which are non-motile to those which are motile. And some bacteria, if I have a bacteria like this, another one like this, another one like this. So if you find the flagella is at one end, we call it monotrichous. Monotrichous means it has a single flagella at one end. And an example of a bacteria with such a flagella, we have what we call Vibrio cholerae. Vibrio cholerae is one of the example of a bacteria which has a single flagella at one end. And you can find other types of bacteria with flagella, one single flagella at both ends. And this one we can name it Amphitrichus. This is amphi means two-sided. Amphitrichus. And the bacteria which have one flagella at both ends, one of the example is Alcarigens fecalis. Alcarigens fecalis. And also some Spirillum species. also have what we call amphitrechous arrangement. Then there are those bacteria whereby you find they have more than one flagella at one end. For example, you find a bacteria with many flagella, more than one flagella at one end. And these ones, we call them Lophotrichus. Lophotrichus. And here an example still we can give Spiridium species, which possess more than one flagella at one end. Then lastly, you find some bacteria with flagella along the bacteria. If this is the bacteria, you find a bacteria with flagella all over, surrounding the whole, all over the cell wall. And this type is known as peritrichus. Peritrichus comes from the word perimeter, meaning surrounding. So we have peritrichus. So you can appreciate that every word ends with suffix trichus. Trichus. Mono meaning one, amphi meaning two-sided, lopho meaning more than one at one end, then peri means all over the surface. And examples of bacteria with the peritrichous arrangement, here we can see bacteria like E. coli, bacteria like Salmonella species possess flagella all over the surface of the cell. And the most function of the flagella is locomotion, or what we call movement, because they contain the mitochondria, which enables to provide energy for propulsion, whereby they are able to move within their media where they are growing. So this is what we can talk about flagella. And don't forget that flagella originate from the cell membrane. These guys, they originate from the cell membrane, not the cell wall, but the cell membrane. So number 4, we are going to talk about is the pillars or the pili. When there are many, we call them pili. Pili, or you can call them fimbri. So this is the 4th part of the outer membrane, outer parts of the bacteria. And these pili, which we are seeing them surrounding all over the surface, are what we are calling, and they are more in gram-negative than gram-positive. And these pili originate from the cell wall. For them, they originate from the cell wall. That is the pili or the fimbri. And these pili are short. They are short, hair-like appendages. Hair-like appendages. And their function, major function, is adherence. They provide adherence of the bacteria to the surrounding. Adherence either to the surrounding or to the other bacteria. So for a bacteria to adhere or to interact with the environment where it is, it needs the pili or the fimbri. And it can use them to adhere to the surfaces and to adhere to other organisms. And another thing, there are those which have been modified into what we call the sex pillars. And these sex pili, they enable sharing of the genetic material by the process known as conjugation. So the sex pili, they enable sharing of the genetic material, that is the DNA, by the process known as conjugation, whereby the plasmids and the DNA, like for example, the plasmid can be shared to other bacteria by use of the sex pili, which can confer resistance in the surrounding. So this one marks the end of the outer parts of the bacteria, whereby we have the capsule, we have the cell wall, the flagella, and the pili. And we have seen their major functions. So as students, you have to master all the functions of these parts, which constitutes the outer part of the bacteria. So after appreciating the outer part, now let us end with the inner parts of the bacteria. And among the inner parts, we are going to talk about this is the parts which we find in the cytoplasm. And within the cytoplasm, we are going to see different parts like the plasmid, parts like the ribosome, parts like plasmid, the inclusion bodies, and so forth, as we're going to discuss with you. So we are beginning with the inner parts. So these inner parts, the first one is the cell membrane. And this cell membrane, we said you can call it plasma membrane, which is denoted by the red part. And this cell membrane always invaginates into what we call a mesosome. This is a mesosome, which is an invagination, and it is made up of a phospholipid bilayer. Phospholipid bilayer. And this phospholipid bilayer has what we call the hydrophobic tails and hydrophobic heads, hydrophilic heads. So it has the hydrophilic heads and hydrophobic tails. So this phospholipid bilayer enables interaction, or it enables movement of materials from inside to outside of the cell. So the major function of the cell wall of the bacteria is to enable or to regulate movement. So it is responsible for regulating movements of materials in and out of the bacteria. So being a phospholipid bilayer, it enables certain materials which are lipid soluble to enter and those which are water soluble not to enter into the bacteria. That's why we are saying regulation. And this cell membrane, it is the one that encases the cytoplasm. And we are saying all these materials are found within the cytoplasm. So after looking at the cell membrane, number 2, we can talk about a mesosome. Mesosome. And this mesosome, we are calling it is an invagination. It is an invagination which is found on the cell membrane. And this invagination enables different functions like respiration. It enables respiration of the bacteria. It enables cell division, that is by binary fission. And these are the major functions of the mesosome: respiration and cell division. That is what we can see. So this one means the mesosome. In eukaryotes, we have mitochondria. So this one acts as like a mitochondria, because we know in mitochondria, that's where we find respiration. So the mesosome is being used as a mitochondria, which is an invagination. So number 3, which we can talk about under inner membranes, we can discuss about the ribosome. And these ribosomes, they are made up of the large subunit and the small subunit, whereby the large is known as the 50S ribosomal subunit. The small is known as the 30S ribosomal subunit, and all of them make what we call the 70S. So this ribosome, the smaller one is this. This is the large. Then this is the small subunit, which we are talking about. And within the ribosome, it interacts with the messenger RNA. And it is at this place which acts as the machinery for protein synthesis. So this one acts as the machinery for protein synthesis. And within the prokaryotic, like the bacteria, these ribosomes are found freely. We know in the eukaryotic cells, the ribosomes are bound to the rough endoplasmic reticulum. But within here, they are all over freely within the cytoplasm of the cell. And their major function is protein synthesis. From there, let us talk about another internal part, which is known as the plasmid. And this plasmid, it contains the bacterial DNA, but this bacterial DNA is different from the chromosomal DNA. They are separate. And this DNA within the cytoplasm, and we can appreciate that this plasmid, this is what we are calling the plasmid, it is circular. This plasmid is circular, and it is the one that carries what we call the DNA, and this DNA contains what we call virulent factors. And these virulent factors include like antibiotic resistance, and other virulent factors. So this plasmid, when it is shared, because it can be shared by the bacteria from one bacteria to the other via the sex pairs. So when a bacteria acquires a plasmid with a specific virulent factor like the one of antibiotic resistance or maybe acid resistance, it can pass it on to another generation, and all of them can end up becoming resistant to some types of antibiotics when taken for all. So, and this plasmid is being used in genetic engineering to enable manufacture of different phages which can be used to prevent antibiotic resistance in our organisms when we are treating these bacterial infections. So this is what we can talk about. A plasmid is a circular DNA. It contains a circular DNA which carries virulent factors that enable bacteria to survive in harsh conditions. That is what we can talk about the bacterial plasmid. Then we can also talk about the DNA. Can talk about the nucleoid or the chromosomal DNA. And we said the chromosomal DNA is located in the region known as the nucleoid. So number 5 is the nucleoid. And this nucleoid contains what we call chromosomal DNA. And this DNA is single-stranded, and it can be circular in nature or like long strand. But the nucleoid, we said it is the region where this DNA is found, and this DNA is responsible for, it is the one that controls cellular activities. Activities like cell division, activities like, so we are seeing activities like cell division, respiration. All these are controlled by the chromosomal DNA, which we said this DNA is different and distinct from the one found in the plasmid, because the plasmid is the one that confers virulence factors. But this one is just to control all the cellular activities. And this is the chromosomal DNA we are talking about. These are the DNAs found within the nucleoid. And we know that this DNA lacks the nuclear membrane. That's how it differs from the eukaryotic cells, because in prokaryotes, they lack what we call the nuclear membrane. That is what we can tell you. So, as we are concluding, we also have what we call the cytoplasmic inclusion as number 6. And this cytoplasmic inclusion, for it, they act as the space of storage. Storage of cellular materials are stored within the cytoplasmic inclusion so that they can also be able to provide energy and other nutrients within the bacteria. So that is what we can talk about cytoplasmic inclusions. Then, apart from that, the last one we can discuss with you is the endospores. Presence of endospores. These endospores, they are non-dividing, so they do not undergo division. And these endospores, they provide what we call protection They are responsible for protection. So they are non-vegetative. And for them, they confer the bacteria protection factor against harsh conditions. Harsh conditions like high temperature, like change in pH, all these ones, and also even against chemicals. Because these spores, they are very heat stable. They are heat stable, so they can make the bacteria to survive in a hot environment. So, and even against chemicals like disinfectants. So if you are trying to disinfect a bacteria which possess endospores, which we named them here, and the example of bacteria which has the endospores we saw like Bacillus anthracis and Clostridium tetani. So Bacillus anthracis and Clostridium tetani, they possess these endospores. Bacillus anthracis and Clostridium tetani and other Clostridium species. They possess these endospores that enable them to survive in harsh environment like high temperature environment. So meaning they make them to be thermophiles and also against disinfectants. So this is what we can discuss with you, where we have seen inner parts, we have the cell membrane, then we have all these ones that are found within the cytoplasm. That is, we have the cell membrane, the mesosome, ribosomes, the plasmid, the nucleoid, which contain the chromosomal DNA, the inclusion bodies, the cytoplasmic inclusion, and the endospores. So this is what we can discuss with you in this session. We are always glad to serve you. And please remember to like the video if you have enjoyed and share to many others. Thank you.